Our next speaker is going to be Yael Weiss. Um, so Yael is a drug developer, and she runs Mozi, which is a company that is trying to move and translate therapies for rare neurodevelopmental disorders into the clinic. And she has been mentoring all of the folks in the rare neurodevelopmental space, like me, about how to start thinking like a drug developer. And so she's going to, I think, give a presentation about that today. So Yael, I'll let you take it from here.
Thanks, Eric. And hi, everyone. I hope I don't lose half the audience on this topic, but it is really important for us drug developers to work with academics on taking programs from the bench to the bedside, as everyone likes to say. And the reason that Eric invited me to give this talk is that we have a lot of experience working with academics and taking work that was funded, and maybe I'll move my slide, work that was funded by patient advocacy groups, working with academics and taking these programs from the basic research stage into IND-enabling studies and into the clinic. We've done this with the Pitt Hopkins Research Foundation, and I'm happy to tell you that our IND for gene therapy for Pitt Hopkins was just accepted yesterday by the FDA. We're working with the Weiss Foundation on a gene therapy, collaborating with the Hebrew University, and with the Coalition to Cure CHD2, collaborating with the Weizmann Institute. So we do have quite a few, hopefully, success stories of taking programs from an academic lab and into the clinic, but we've had a lot of learnings along the way, and I wanted to share them with you. So what will I cover here? What is translational versus basic research, which I'm sure I don't have to tell you, but I want to start with that. And then what is the path from a lab into the clinic, and what do we do in the background to make sure that the regulatory agencies, whether it's the European, the Australian, the American, wherever we are filing our clinical trial application, what they want to see and how important it is that you know what they want to see. And then I'll talk a little bit about the boring but so necessary details, because if we can work with labs to prepare for the IND or CTA filings ahead of time, it will save us so much time and enable us to get to the clinic faster. So when I asked ChatGPT what the difference is between basic research and translational research, it gave me a very nice definition. Fundamental knowledge is basic research. Something that is needed to take into the clinic is translational research. But in simple words, basic research is understanding the mechanisms, HMT1, what does it do, how does it do, when does it do it, and then taking this knowledge and turning it into a drug. That's a whole different level of research, or different area of research, that is translational research. And it's not for writing a grant, it's not for a publication, it is to match the sense of urgency that the patients have, and in this case, the Kleefstra, the I-Define and the Kleefstra community, and really needs to be thought about in advance when you're conducting your experiments in the lab. So what is actually the practical difference? The practical difference is how you plan your experiments and how you report them. And it's really important that planning is pristine and reporting is even more pristine. And I'll talk a little bit about that. So I wanted to start with this, which I share in many of my talks. This is a target product profile. This is what we use when we start thinking about what our drug is going to look like and what our label for the drug will look like. All of you open a box of pills, and there's a long piece of paper that is folded and nobody ever reads, but that's the target product profile. That's what we're aiming to. How do we populate this paper inside the insert of the drug package, and what will the drug look like, and what are we asking to be approved by regulatory agencies? So the first thing is the mechanism of action of the drug, and what is it going to be used for? And then who is it going to be used for, and how is it going to be used? And what I want to make note of is there's very little basic science in the target product profile. It's mostly clinical. So even though basic science is the one that will lead to the drug and the clinical studies, very little of it is mentioned in any future commercial drug product. However, everything on here will be driven by findings from basic research and translational research. We obviously will hone in on the patient population by getting data from you. Does it work in pediatric in a young mouse, in a prenatal mouse, in a postnatal mouse day 0, in a postnatal mouse day 21? Will this allow us to understand if we treat only a pediatric population, or can we see any hope for treating adult patients? So your experiments will drive those decisions. Dosing, of course, as Neil just showed, you can show dose responses in your cell models and in your animal models. And of course, that will apply to how we determine the dose in human beings later on. And another thing that's interesting is that whoever wants to see the target product profile, whether it's payers or regulators, they want to see what our target is, and then what is our aspirational, like what is the best case scenario, and then if we don't meet the target product profile, what is the minimal profile that we can achieve for this drug? And that again will be driven by experiments done in your labs, but also by the IND enabling studies and the clinical studies. So what differentiates data preparation for an IND? Really, really important. The protocol is predefined to the nitty-gritty detail and cannot be changed. And if it's changed, then why has it changed? So for example, we've had an instance where we required a P0 injection in a mouse, and the student was sick. I understand, students get sick, it's okay. You can't force them to come to the lab when they're sick. However, they injected in day P3, and that skewed the results completely. So what do we do now? We have to report the P3 injection, and it wasn't as successful as the P0. And we have to amend the protocol retrospectively to say the injection took place in P0. So I know it's impossible to ask people to be perfect and not go on vacations, etc., but you need to understand that every single deviation from a protocol has to be reported when you're filing the IND and given justification. So we learned the hard way. I hope our experiences will help others learn how to do it and to pre-plan for whatever emergency or whatever situation may happen where you can have others step in and do work to adhere to the protocol. Everything is reported, as I said. Every relevant piece of data has to be reported. So successes, failures, everything has to be reported. So we need to take this into consideration. There's not like, oh, this experiment, the p-value doesn't look great and I can't put it in the IND. It has to go into the IND because the regulators want to know not only what went well, but also what didn't go well. And as I said, it has to be detailed and accurate. Just for those who don't know, I'm going to briefly describe a little bit about the IND process, and happy to go quickly through it, but if there are any questions, I can expand if needed. So I'm talking about the US regulatory agency because that is what American companies usually go to, but we also obviously file in Europe for European clinical trials. But in the US, there are two divisions, CDER, which is the Center for Drug Evaluation, and CBER, the Center for Biologics. CDER sees, as Neil mentioned earlier, small molecules, and the antisense oligonucleotides go to CDER. They're not considered a biologic, they're considered a small molecule. And then CBER is the Center of Biologics that sees all the products that have a biologic derivative. So it could be blood products, vaccines, gene therapy, cell therapies, etc. So for example, in our case, we just submitted a gene therapy. It was submitted to CBER, reviewed by the rare disease unit and neurology unit, but CBER was the one that was responsible for the review. The regulatory pathways are the same. There's an investigator IND in case an academic institution is running the study. There are emergency use INDs and then treatment IND, which is what companies usually file. What does the IND package include? A lot of stuff. And just to give you an idea, an IND package could be 2,000 pages long. This is why everybody wants to use AI to read them, because they're so long, it's impossible to read. But the requirements are to submit information and very detailed information on everything that's in this slide. And I circled the two places where the basic research and the experimental translational research actually has to be described. One is in the pharmacology and toxicology part of the IND, and the second is in the investigator's brochure. So actually, when we put together the clinical trial, we prepare a brochure for the PIs, the principal investigators, so that they understand how we got to where we are with the drug. So we have to actually summarize very succinctly everything that was done in order to get the drug to this stage of clinical trials, and so that they, if they have questions from patients, questions from IRB, so they can explain where the drug came from and what experiments were done to get it to clinical trials. This is the core of an IND package. Module 4, it's everyone's nightmare. It's a very long module. It's very detailed, and it contains all the basic research that was done on the drug, all the translational research, and the IND enabling package, which is usually toxicology and pharmacology. And it's described in detail in module 4, but also in module 2, which is the summaries of all the modules for the reviewers to start reviewing the package. What is the purpose of module 4? It's basically a safety assessment. The reviewers want to know what the risk benefit of the drug is, so you have to explain everything that led you to understand that why the drug has potential to be effective, but also what are the safety margins for the drug. So what is the therapeutic window that you're actually working in? Even though it worked in 10 different doses, if the 5 top one were toxic, then you're left with only 5 doses. So this is where you explain the rationale of how you chose the dosing and what the doses, how the doses from the preclinical studies translate into the human studies. The format is harmonized. I'm happy to provide a reference if somebody's really, really bored going on vacation, sitting on the beach, and wants to read all the regulatory documents. There's a document that describes exactly what goes into these descriptions and into the modules that we have to submit. And I'll go through them very briefly just so that you understand what we ask you to do and why we ask you to do what we're asking. So the components, the high-level components of Module 4, and I bolded the 2 that are relevant to academic researchers. One is the study reports. So that's the study, the reports in which we submit all the data that comes from the labs, and the pharmacology studies, which is basically the experiments that showed how the drug affects the body, what the positive effects are, what the negative effects are, and what the intended mechanism of action is. Obviously, there's a lot more in there, but I think that most of the work that is done in academic labs is covered in study reports and pharmacology studies. So what do we do when we actually start preparing for filing an IND? Is we actually have to take all the data that comes from your labs and reformat it into an FDA compliant package. So the more compliant it is when we get it, the less time and energy and effort and cost it will be for us to actually put it into the package and submit it faster. So we have to create detailed study reports, and I'll talk a little bit about the details that have to go into the reports. We have to author the summaries of these reports and establish a clear narrative. What is the story? If the work was done in 5 different labs and one did the cell work, the other did the mouse work, one did electrophysiology, and the third did behavior, the fourth did behavior, how do we take all this together and create a story that will make sense and address the need to understand how the drug works and why we think it should be taken into population X or population Y. So this is a laundry list, and this is the most critical list. So when we design or devise a protocol with an academic collaborator, this is what needs to be in the protocol. And in most cases, it's way more detailed than what a student in the lab would do. And the main issues that we encountered is that many times the work was done 2 or 3 years ago by a grad student or a postdoc that left the lab a long time ago. And now you have to go back to their notebooks and try to reconstitute what really happened to provide us with the details that we need. So if you know in advance what we need, maybe it will be easier for you to predefine and to make sure that this data is available even when somebody leaves the lab and moves on to a different country or a different career. So the degree of the detail is to understanding obviously what the test article is and the controls are, but it's a very detailed description of the methods, a very detailed description of the test system, including number of animals used, sex, body weight, source of supply, species, strain, substrain, age, procedures used for identification. Blinding is really critical. How did you blind the study? If it was blinded, was it blinded by 2 different people conducting different sets of experiments, or you didn't know what the genotype was and you genotyped only after the experiment? Really important to know how blinding was done. Obviously dosage, dose regimen, route of administration, etc. And if anything affected the quality of the data, there was anything like somebody couldn't come on day 0 and came on day 1, you have to justify it and include it in the report. List continued, the report has to be signed and dated by the study director, and then corrections will be in the form of an amendment. So everything needs to be formalized and signed so that the regulatory agency knows that whatever was included or excluded from the report was done on purpose, was done in an informed way, and that we're reporting what really happened. I included here our FDA correspondence. I think it was really interesting when we went to our pre-IND meeting and we said, this is the data that we have. Is it enough? They came back to us and this is what they said. All the study reports, please document all attempts made to minimize potential study bias. So that's really critical for them. Inclusion of appropriate controls, randomized assignment of animals to study groups, appropriate staggered dosing of animals across groups. So if you dose the controls on one day and the test article on another day, they need to know that, and will this affect the results and the quality of them? And obviously the blinded assessment of the study parameters by a qualified personnel. And if this is not done for the completed studies, please explain why and discuss what was done to minimize possible study bias. Animal deaths. Everybody comes in the morning and finds a mouse dead in a cage, usually already liquefied. What do? You still have to send this animal to perform a comprehensive pathology analysis, histopathology analysis, because they want to know if it's drug-related or not drug-related. You don't always get the answer, but you have to do it. So this is really important to know. We have had cases where mice were found in the morning or a day later or a few hours later, and the student thought it's worthless to even test it now, and the mice went to wherever dead mice go. So the report should not be limited to a prospective limited protocol on all protocol amendments in a detailed methodology. Should include but not be limited to a prospective protocol. So as I said before, you have to design everything in advance. You can't improvise as you go based on the results. If this is what you decided you're going to do, that's what you're going to do. If you do want to change something, it has to be in a form of an amendment. You have to give a detailed description of the study design, the test system used, all the details of the procedures to the animals, and then we need the raw data. It's not enough to get a really nice graph with p-values from you. We need the raw data because we deposit the raw data in our files, and the FDA may go back and analyze it. They may not trust what we submitted. They will go back to the raw data and recalculate in certain times. So we need the raw data. And something to remember, because that's the thing that we usually have the hardest time getting if someone moved on from the lab and is no longer there. This is my extra credit. This is the document. If anybody wants to read it, it's called the M4S, the CTD safety document. what I wanted to talk a little bit about at the end is a proof of concept. What do we see as a proof of concept package? Obviously, it's on a case-by-case basis. What is the disorder? What are we trying to treat? What is the modality that we're using? But ideally, we'd like to see data from cells and from a mouse model if one exists. It's not mandatory. In some cases, it's not possible to treat a mouse with a human-targeted drug. But if there is one, that's great. Cells, either isogenic or patient cells representing the most common variants, organoids, if there are organoids with a phenotype, that's great. A mouse model, obviously it's not human, but it is well accepted if the model is relevant. Does it represent the disorder? Yes or no. Does it have to represent the disorder? Not necessarily. In some cases, we just want to show that we are able to fix something. Doesn't necessarily have to be the same as the human condition, but to show that the drug distributes and has its effect in certain tissues that we're targeting is helpful if we can see it in a mouse. And then the question is P0 enough? Do we need P21? We submitted the package for Pitt Hopkins just on P0 data. We didn't have later data, and it was well accepted by the regulatory agency. I'm not saying this is the rule, but obviously it's not impossible. And of course, biomarkers are really important. I'll give a quick example of Pitt Hopkins and what we had when we licensed the program into Mozzie. So Pitt Hopkins, if you've never encountered it before, it's mutations, haploinsufficiency in TCF4, which is a helix loop helix transcription factor. Similar to HMT1, it's a de novo loss of function and very similar presentation to Kleefstra, less seizures, but a lot of other similar characteristics. What we had when we licensed the program is proof of concept of in organoids from Professor Motri's lab at UCSD. And he showed in the organoids that he can express 100% of TCF4, so restore the levels of TCF4 expression, reverse electrophysiological defects, and restore the organoid infrastructure. And then we took the gene therapy construct and put it in a different lab in a mouse model and showed that we are re-expressing the protein in the mouse brain and we are restoring behavior and dendritic spine length. So this was the non-clinical, preclinical package that we had to show proof of efficacy for Pitt Hopkins gene therapy. This is the publication. I won't go into details, but this is the organoid work, and this is the mouse work that we did. So it includes the behaviors on the left-hand side and the dendritic spine length on the right-hand side. And this is the data that we submitted to the FDA. I'll stop here because Eric asked me to talk a little bit about clinical studies as well, but if there are any questions on the non-clinical part, I'm happy to answer them now. I'm going really fast, so I hope I have some time for Q&As.
Hey, Yael, if you want to do those questions now, I don't see any in the chat currently. I had one, which is there's always a challenge with INDs because they're not public, and so it's not easy to sort of look up examples. Like for me, if I can see an example, like that really helps me understand
we're happy to share our IND. It's not a problem at all.
Okay.
There are companies now that are sharing their INDs, not a lot, but we are happy to share ours.
I've heard of a few that are posting them, yeah. So there was even one that posted theirs, I think, on the internet.
I don't know if we'll post ours, but definitely we'll share it.
Share them in confidence, yeah, at request, I guess is what you're saying.
It's just, I don't know that you actually want to read an IND. It's so long. But
well, I was looking up when you were saying they're 2,000 pages. I looked up the Lord of the Rings trilogy is 1,500 pages. So it's longer than the Lord of the Rings trilogy.
It took 14 people for us to write the IND and publish it and quality control it. So it's a heavy lift. But I think actually the interesting things are not the IND itself, but more the questions that you get at the pre-IND meeting and how you address them in your plan moving forward. So we got a lot of questions about the basic research, and we had to go back and conduct some additional experiments. And then that helped us plan our toxicology studies. And then the questions that we got now when we submitted the IND and we had to answer before the IND was cleared. I think these are the most important ones, because then
Hmm.
you know what the emphasis is of the reviewers when they look at your package.
Very interesting. Okay, so I guess the sum up is someone can be in touch with you if they'd like to learn more about kind of
Yeah,
your IND experience. Okay,
sure.
wonderful. I think we can go ahead and move on to the next segment then.
So the next segment is short. Eric asked that I talk a little bit about what happens after we have proof of concept, and then we start planning for clinical trials, and we don't wait till the IND is cleared, obviously, because in the IND you have to already file your protocol. So you have to have a full protocol. You know what your endpoints are, know what the visits will encompass, and all that. So we start planning as we finalize the proof of concept in an academic lab. We already start planning for clinical trials. So we're assuming that we're working on disorders that have very limited previous clinical trial exposure, and most of the therapies that we work on, at least we hope, will be disease modifying. So what are the challenges in a disorder that hasn't been in the clinic, and maybe has been in a clinic for symptom relief, but not for a disease modifying therapy? What are the endpoints that you will be using? Are you taking endpoints that somebody else has used for a different disorder and just validating them for your disorder? Or are you creating new endpoints based on what you know about the disorder that you're working on? So the most important thing is to hope that they will be sensitive enough to measure change in a clinical trial. The studies are small. Our Pitt Hopkins study will have 12 patients, 2 cohorts. One will be an adult cohort and one will be a pediatric cohort. We were very lucky. We were able to convince the FDA that we can do a single dose study, so we're not doing a dose escalation. So the studies are tiny. So the samples you can get from them, very small numbers. Obviously, we're waiting for CSF. We'll have only 12 samples of CSF. So how do we decide on the number? How do we work with the regulators to accept the number? Will the study be open label or sham controlled? For gene therapy, there is acceptance now for open label studies. We were asked to do a sham for our first pre-IND meeting, and we pushed back and said it's not ethical to run a sham in such a fragile population. And so we ended up doing an open label study. But open label study, the burden of proof is even higher because you don't have a placebo to compare to. How do you collate all the input? You get input from the patient community, you get input from clinicians, and then the regulatory input on top of it. And from this, you have to come up with a study that will make sense and will hopefully yield good results. So that's a challenge as well. And then, do you have a biomarker? Obviously, in most cases, we don't have access to CSF before the study. And we're relying on blood biomarkers, and we don't know the correlation between a blood biomarker and activity in the brain until we have some clinical trial to show if there is a change before and after. And of course, the regulatory, which we talked about. What we do at Mazi when we start working with a new patient community is we start with a survey. So we do a caregiver survey, which is an online survey, and as many people as want to participate in can participate and give us some idea of what a day in their life looks like, what bothers them most, what they would like to see changed. After we have an idea of what the direction is of these surveys, we conduct in-depth interviews and get a very qualitative patient narrative. And this helps design the natural history study. And then out of all of this, we build a conceptual model that will lead to the clinical endpoint selection. So this is the conceptual model for Pitt Hopkins. A lot of things were brought up in the interviews, but you'll see the green blobs next to certain bullets. These are the top 5 ones that came up in our interviews and in the online surveys. And these are the ones that we focused on for endpoint design. So speech delays, motor delays, feeding difficulties, constipation, and respiration in the case of Pitt Hopkins. So these are the 5 top areas that we will focus on as our endpoints. Don't forget, we're running a phase 1/2 study, so it's not a approval study. So these endpoints are now exploratory. They're not primary or secondary. And based on what we see in the first 12 patients, hopefully we can learn where to hone in and what the measures will be for the approval part of the study. And this aligned with a publication that came out from a European consortium on Pitt Hopkins. So you can see that what came out in our interviews was also highlighted in this publication of a smaller patient population and a less formal process of surveying the community. So these were the areas that we chose to focus on. And I'll end with just saying that obviously, because we start so early working with the patient communities and with academic labs, the goals have to align. The goal is filing an IND, getting into a clinical trial, and getting a drug approved, and the sense of urgency needs to be matched. The sense of commitment needs to be matched, and an understanding that it's not a grant proposal or a publication, but really people's life in stake here. So I think I'll end here and take any questions. And if not, then Neil has more time for his questions.
Thanks, Yael. I think that this was really good, and I can totally see. I think this is recorded, and I think everybody we fund, I'm going to send a recording of this so they can watch this when they're thinking about how to develop their projects. And I thought it was just really interesting points, like you have to have all of the raw data, you have to keep it forever, and you have to have it, and it's going to be asked for. So there was actually a question in the chat earlier this morning, and I realized you'd be a really good person to ask this to. So and it said, much of the research is on young patients with still developing young brains. How will this research and or medications affect an older patient in their 30s and older? Will there be anything to help a person of this age improve their cognitive abilities?
So I don't know that I can fully answer this question. And it's interesting to see what different companies are doing now. Some companies like Jaguar working on SHANK3 and I think also Capsida actually restricted their clinical trial to a pediatric population because of the notion that the effects could be better in younger patients. We decided to start with an older cohort. So our first cohort is 12 to 25. First of all, because since it's the first time this drug is going into humans and the blood-brain barrier in young people and the brain is developing in young people, they may respond differently. So we wanted to see safety in adults first. And also I'm hoping that we will see improvements in adults. And then we will go down to the younger age. So different companies think about it differently. Angelman Foundation, for example, will enroll, I think, 2 years to 40 years because they really want to cover their whole population. So in some ways, it's the company deciding on what the best way to get to patients is, or where are the highest chances of seeing improvement, versus really wanting to cover the whole population and hoping that older patients will see benefit as well. And time will tell.
Yeah.
The more studies we have, the more data we have, the more information we'll have on this.
Yeah, the way I look at it is we just don't know, and that's incredibly exciting. That we don't know yet, and we're going to be finding out in the next coming years. So there was a follow-up question from Jeff. Just he wanted to ask, how would endpoints factor into the age range? Would that maybe change the endpoints that are used or the endpoints you think about?
Well, some of the tools that we use to measure behavior and speech need to be adapted based on the age, because that's why natural history is important. Also, you need to know what an average patient at a certain age can do or cannot do, and then be able to measure change. So we spend a lot of time looking at caregiver reporting, looking at the different, Vineland and all the measures that we're using, and to make sure that they're relevant for the patient population that we're testing. Yeah, you have to have measures that are relevant for the age that you're treating, obviously. Yeah, so we will see. We have several exploratory endpoints that we will learn a lot from and we added videos, which we think are very important. They're
Mhm.
not accepted by regulatory agencies yet, but we hope that with time and with evidence that videos are done in a very regimented manner, that they will be accepted by agencies as well. And I think that's really important.
And will those be at home videos that are done by the families? Okay. Wow. Yeah,
Yeah,
that is a move.
but you have to follow a pro. You can't just
Yeah.
decide, "Oh, today my child is having a horrible
Good
day.
fit.
Let
Yeah.
me
All
record
right. Yeah.
the worst day of his life, and then after treatment, find the best day." It has to be very, very regimented and
Yeah.
very clear that you're following the protocol as well,
Yeah.
which may
I
be
had a
a little bit
question.
challenging to families, but I hope they will be able to do it.
I had a specific question that popped up when you were showing the exploratory endpoints for this study for Pitt Hopkins, and one of them was constipation. And constipation and GI problems is a huge issue for KS as well. And how do you guys think about this is a treatment that's going to go into the brain? How do you guys think about how that would or would not be expected to show up, right, in
I don't
constipation?
First of all, the treatment does go back into the body, so it does leak back. So there is systemic exposure. Whether it's enough or not, we don't know. And we really don't know if the reason for the constipation is central, peripheral, microbiome. I mean, there's so many theories, right? So we added it because it's such a pain point for the families, and we want to be
Mhm.
able to measure it. So we have what we call poop diaries in our
Yeah.
study, and we looked into potentially including more invasive procedures to measure gut motility and whatnot, but we decided not to at this point, at least.
Yeah, and I think if you find in your work that it does affect GI, right, that would be a huge potential impact. It kind of shows how all these disorders are going to benefit from the different trials that are done for each one. Because if you guys find that you actually can affect it, then that is informative for us. That
Yeah,
yeah, in fact, you can affect GI by treating the CNS, and of course, like you said, there will be distribution beyond the CNS. So
yeah.
um, yeah,
I don't know that we can get a drug approved just based on reversal of constipation, but it's definitely a
yeah,
component of a milieu of issues that we want
yeah.
to show that are improving.
How does the FDA look at those primary endpoints versus secondary endpoints? And at the end of the day, how are they going to weight? If it mostly helped with just constipation, let's just for the hypothetical, and that's like a kind of a secondary endpoint, then
Yeah.
I'm assuming that's not enough. But do you have a sense of how they're going to balance these weights,
I
particularly
don't
in
know
rare
that it's
conditions?
necessarily the FDA. It will be the payers because a
Okay.
payer can
Yeah.
look at this $1 million gene therapy and say, gosh, am I going to pay $1 million just to fix
Yeah.
constipation? Probably not. So
Yeah.
the
Yeah.
value proposition has to be more than just I think the FDA may approve it or may not. I don't know. I don't know. But it will be hard to get reimbursement for expensive gene
Yeah.
therapy that all it does is reverses constipation.
Yeah. It's a good point.
Yeah.
Okay. Those are all the questions. And we are on time. So I think we will stay on time. Thanks so much, Yael. Really
Thank
appreciate the
you.
presentation, everything you've done for all the rare disease groups.
Thank you.